Series today is Professor Uma Ramakrishnan. Professor Uma Ramakrishnan is a molecular ecologist, professor and senior fellow, India Alliance at NCBS. She has spent the last 18 years understanding the conservation genetics of Indian tigers. Using primarily non-invasive samples, she and her team have tried to understand population connectivity and the fate of small and isolated populations. She won the Parker Gentry Conservation Award in 2016, the Homi Bhabha Award for Science Education in 2020, the Beacon Award in 2021 from the Society of Conservation Biology, and the Molecular Ecology Prize in 2023. She is a fellow of the Indian National Science Academy and the Indian Academy of Sciences. Uma loves animals, researching India's wondrous natural landscapes, working alongside early career scientists, and trying to communicate research to diverse audiences. Professor Uma will be speaking on Project Tiger, Advancing Tiger Conservation with Genetic Science. Welcome, Professor Uma.
Thank you, thanks for that introduction, and I always prefer these mics because I move around a lot. Hi everyone, I'm Uma. Nice to meet you, and I'm going to talk a little bit about charismatic species, so forgive me. I know you heard a lot about ecosystems and global change from the earlier speaker, but I'm going to give you like just pure eye candy, okay? So I work on tigers, and I work on them from a genetic lens. I'm basically a population geneticist. And basically, we all know that biodiversity, surrounds us, and it's really fabulous. Anywhere you look, there's biodiversity, and it's really beautiful and fascinating, whether you're talking about butterflies or, landscapes like these forests or charismatic species like this leopard. And you'll hear later in the day from Kartik, but this biodiversity is also not equally distributed around the world. So I love maps, and this map basically shows the distribution of vertebrates. The brighter, the hotter the color, the more red, the more biodiversity there is. And you can see, for example, etched in here, mountain ranges, for example, where there's more biodiversity than other places. Okay, thank you. So you'll hear more from Kartik in the afternoon about this. Yeah, great. But I just wanted to point that out before starting. So biodiversity is beautiful, biodiversity is everywhere, but does it really matter? Especially for us sitting here today comfortably in this AC room, does biodiversity matter? So there's a lot of discussion about this, and recently you've also seen, in fact, global movements, right? From people like Greta Thunberg, protests. Biodiversity and the value for biodiversity is becoming almost a political cause these days, and that's because we are losing biodiversity very rapidly. This is the IPBES report from the last time, and it suggests, for example, that there's high rates of species extinction. 25% of species are threatened with extinction, for example. And if you look, for example, at, say, amphibians, it's even higher, but mammals, for example, 25% of species are threatened with extinction. So this sounds very, it sounds really bad that, so many species are threatened with extinction, such beautiful species from across the world. So as biologists, I'm a biologist, what do we do about this? It sounds very much like a, activist kind of thing. But before we go there, I'd just like to show you maps of India from 1700 and 2000. So you can see, for example, that what is like green is completely or to a large extent gone. So basically, we've lost, forest cover has gone from 60% to 9% in these 300 years, and agriculture and villages have increased from 20% to almost 80%. So what remains now are these fragments of habitats, right? Kind of islands where our biodiversity, not all of it, some of it is kind of stuck. So as I said, I'm a biologist, and so the first question we ask ourselves as biologists is what causes extinction? So we're shouting and screaming on the streets saying biodiversity is going extinct, we need to save it, but how do we do this as scientists? So the first question we ask is, why do populations go extinct? This has been worked on for a long time, and this is an example of data from butterflies, which basically shows that the probability of extinction depends on population size. So the larger the number of butterflies, the lower the chance of them going extinct. There also seems to be like a threshold size, roughly like 100, population size of 100 seems to be pretty important. Higher sizes have generally low probabilities of extinction. Another thing which affects extinction is isolation. So this is slightly harder to read, but this basically shows the number of patches and how isolated those patches might be. So if you have many connected patches, the time to extinction is very long, so probability of extinction is low. But if you have few isolated patches, the time to extinction is quicker. So basically, small and isolated populations tend to have higher probabilities of extinction. And what about genetic variation? Does this matter to extinction at all? Well, this data again, old study on butterflies shows that when you have, sorry, I'll come this side. When you have higher, this is on the x-axis you have heterozygous loci, or you can just think of it as a measure of genetic variation. When variation is high, the probability of extinction is really low, but when variation is low, the probability of extinction is high. And the reason for this, there's two reasons. One is small populations which tend to have lower genetic variation are also subject to stochastic processes which by chance will fix or result in high frequencies of not such good mutations. And the second is that small populations also have inbreeding, mating between relatives, which exposes these bad mutations and causes inbreeding depression. So in summary, extinction then is a function of population size, isolation, and some kind of an index of genetic variation. So what has this got to do with tigers? So it's like I told you in the beginning, this is all eye candy, right? So this is just a nice picture of a tigress in Bandhavgarh. And basically what we know about tigers is that this was their historic range. There has already been extinction. So here, for example, tigers are extinct in the Caspian area. Tigers are extinct in South China. Tigers are extinct in Java. But overall, while there still are a lot of tigers in India, 70% of the world's tigers live in India. We can understand more about process of extinction, maybe by studying tigers. So basically what I'll do today very quickly is talk about, so what are the populations of tigers then, right? What is the unit, the evolutionary unit of tigers in the Indian subcontinent? What are these landscapes where tigers live, and what allows for them to be connected across these protected areas? What potentially impedes connectivity? What happens in small and isolated populations? We already have them, and we have now two examples of drift and inbreeding. And then basically, how do we move forward? What are we doing now? How are we going to move forward to the future? Okay, so how do we study tigers? So basically, we walk mostly through protected areas because that's where the density is high. We often work with fecal samples because they are very plentiful. Tigers mark their territory by pooping and spraying urine to mark their territory. And basically, we can collect fecal samples. The fecal samples, because they go through the intestine, they have tiger epithelial cells, right? And we can try to get DNA from these fecal samples, tiger DNA. We also increasingly work a lot with shed hair and saliva because this fecal DNA has many other things in it. It has bacterial DNA, it has prey DNA, and the proportion of tiger DNA is very small. Whereas if you work with shed hair, for example, an animal is sitting, you go and pick up hair afterward, you have very low amounts of DNA, but it's mostly tiger DNA. So I'll show you some of that going forward. So of course, you all know this, but basically, this is something which we can only do recently. So for a long time, many years ago, we learned about genetics and the concepts of genetics from people like Mendel and Darwin, but it was only the structure of DNA in 1953 which allowed us to think about how can we read DNA sequence. And very critical to this effort was methods developed by Sanger, which allowed us to sequence and read DNA. But basically, another key innovation was polymerase chain reaction, which allowed us to copy DNA and actually then read it. So now when we're talking about tiger feces, we have very little amount of DNA. So we need to copy it and then we need to read it, right? So PCR allows us to copy it, but it was this huge change in technology, next generation sequencing, which allowed us to get much more data for less cost, and basically the cost of sequencing has dramatically declined, and this is what has allowed us to basically sequence tiger genomes and understand more about tiger genetics. So what we did first was to try and understand the distribution of tiger genetic variation across their range, and we collected samples from India, other parts of tiger range, this is the Russian Far East, Sumatra and Malaysia, and we basically sequenced these tiger genomes. Tiger genome is 2,400 million letters long. So we sequenced several individuals, and then we basically used various statistical approaches to ask, do some of these tigers, are they more similar to each other than they are to other tigers? That's what this PCA analysis does, and what it shows you is that yes, tigers from India called the Bengal subspecies relatively closer to each other than they are to Sumatran and so on. This is the same kind of thing, different analysis, but also very interestingly, some individuals, these are from Northeast India over here, seem to be admixed with variation from other parts of Asia. I think the most interesting plot is this one. It basically is a rarefaction analysis showing in some sense genetic variation as a function of number of individuals. And basically what we see is that Indian tigers have really high genetic variation, all of them together compared to other tiger subspecies. But the other thing we also see is that this plot actually shows what's called runs of homozygosity, or long stretches in the genome which indicate inbreeding. But what we also see here is the pink is Indian tigers. Overall, while the total amount of genetic variation is very high, individual tigers in India are quite inbred. Okay? So basically this could be because all of our habitats are now fragmented. We'll talk a little bit more about this later. So basically India does have a substantial amount of the number of tigers as well as tiger genetic variation, and so if tigers are going to survive conservation in India, for their genetic variation is really important. So basically we talked about how isolation in small population size is bad. So what is it that allows tigers to move, right? So we studied this some time ago. In this landscape, that's central India, basically MP, Madhya Pradesh, Maharashtra and Chhattisgarh. And in this case, Prachi, who was a PhD student then, sampled tiger feces walking several tens of thousands of kilometers, and then basically compared tigers from these different populations genetically and tried to understand, given, say I compare a tiger from here and here, what is the landscape which is in between those areas, and how might that be allowing for movement or stopping movement. So basically using this entire landscape, we constructed what's called a resistance to movement surface, which shows us basically the darker colors suggest that those are areas where genes are not flowing, tigers are not moving, whereas the lighter areas are those where tigers are able to move. And what this resistance surface showed us is that overall populations over this large landscape, it is really a large area, are connected, but high traffic roads and high density human settlements like cities are posing barriers for tiger movement. Now while this study was going on, between these two protected areas, this is Kanha and this is Pench. Pench is where Jungle Book was written by Rudyard Kipling. A highway was being expanded. So this is NH7, and this was the highway. So this was in 2016. We published this paper in 2017, and this highway was being expanded from a 2-lane to a 6-lane highway. But because of this research, which basically showed that individuals were moving between Kanha and Pench, and that if this highway was expanded, it became a high traffic road that would become a barrier for movement. Citizens were actually able to put in a public interest litigation, and basically NHAI was ordered to make this overpass for the traffic. So sometimes people ask, what's the point of doing this kind of science? It doesn't really go anywhere, but in this case, it was used in court as evidence for biological movement and basically allowed this underpass for animal movement. So basically, like I said, this DNA evidence was used in court for this mitigation structure. And what's really heartening, these are camera trap images from the underpass, and you can see that this tigress and her cubs, these males, are the ungulates, these are dhole, Asian wild dog, they're actually using that underpass and they're actually moving between Kanha and Pench. And even if you look at it today, genetically, Kanha, Pench and the Kanha Pench corridor is one genetic hodgepodge. It's all mixed up. So that's saying that this allowed for that connectivity to be sustained, hopefully into the future. But if we want tigers to move, so we're saying we don't want them to be isolated, we want them to keep moving across the landscape. In a country of 1.4 billion people, they are going to end up interacting with humans, right? So what do we do about this? So this is just a really nice set of pictures. This is actually a place outside of Bandhavgarh Tiger Reserve in Madhya Pradesh. Camera trap pictures. This is a female and her 3 cubs at 9:15 p.m., 11:30 p.m., 7:40 a.m. and the same spot at 12:45 p.m. So really, this is not hypothetical, really tigers are overlapping with people, and that results in livestock depredation. First of all, that is, a lot of cows or buffaloes are being killed by tigers, and then sometimes, while there is compensation, sometimes there is like dissatisfaction with this, and that could potentially lead to backlash against these animals, which could potentially again become a limitation for connectivity. So we wanted to ask in a kind of a very forensic way. So generally if you ask people, okay, what's happening with this livestock depredation or conflict, it's called human-wildlife conflict, they'll say there is this one animal, it's really problematic, it's coming and killing all of our cattle, okay? Or it is actually attacking humans. And I don't know whether any of you have seen this movie called Sherni. It was pretty nice movie, and there they portray this female tigress whom they're all trying to catch. So we wanted to ask, is this really true? Is there really a problem individual who is causing all the issues? So how did we do this? Well, for that we need to think a bit about animal behavior. So this is 2 cubs, again outside Bandhavgarh. There's a cattle kill here, you can see, and what are they doing? They're like really handling this prey, right? They're handling it. I don't know whether any of you have cats, but you'll see that they actually handle their prey a lot. So when they handle their prey, what do? They actually leave biological material on the prey. So here, for example, we have jugular bite mark. You can see a bite mark here, and all of this hair is actually not the cattle hair, it's the tiger hair. So basically, if you just kind of look at this more closely, it's like a crime scene where you have various types of, you have bite marks, you have lick marks. Cats always lick their prey to clean it, and then you also have a lot of hair and sometimes fecal samples around, but all of this is evidence for who killed that cow, right? It's evidence to understand if we can get DNA out of this stuff, we can figure out which was the individual involved in this kill, and if we sample many kills, we can understand, are there specific individuals who are problem individuals, or is it something different? So that's what we did. I'm just showing you the results. This is just new work, not yet published, but basically we sampled about 200 cattle kills around Bandhavgarh Tiger Reserve, and this shows you we identified about 50 unique tiger individuals, and what this shows you is that on an average individuals were present at 1 or 2 kills. So when you total this, something like 40 to 50% of the population of Bandhavgarh was involved in cattle kills. So this is not, there is one problem individual here which was going to say 7 kills, was found at 7 kills. But overall, this seems to be much more ecological. There's lots of cattle, and so tigers are actually predating on these cattle. So this basically also shows you the distribution of this across space, and there are some pinch points like some areas where there's more conflict, right, more kills are happening, and that's something which we need to explore further. Now, what about the utility of this method as a technique? For example, can I actually hand this over, like I'm doing this at NCBS, right, it's a lab, very well set up facilities. Can I hand this over to a manager who wants to actually figure out what are the individuals involved in conflict? So to do that, we basically analyzed the results a bit more statistically and asked how many times can we actually figure out which individual was responsible for a kill. So basically in 86% of the kill sites, we can identify a unique individual which was responsible. Overall shed hair appears to be much better. This was a surprise compared to saliva or scat. The success rate is highest in winter, so even 4 samples from a kill site were enough to identify an individual involved, whereas monsoon is much worse. This makes sense. Rain washes off the DNA, and so your success rate goes down. So overall, there's a strong signal of season. That is, it's going to be very hard to identify who is involved in the monsoon, but overall, this technique seems to work. So this is something we're working on now. We just got a request from the Maharashtra Forest Department. So by the way, this is just I have genetically identified a tiger, right? But who is that tiger? That I don't know. I don't know the physical tiger. So for that, I need to build first a database of tigers, like an Aadhaar card for every tiger, where I have genetic information on that individual, camera trap photos, so I can go and find that animal later because they have unique stripes. So that's something we're just starting to work with the Maharashtra Forest Department to try to profile individuals first, and then we can try and match who is found at a kill site to which individual it is. Okay, so now I'm going to talk about some work which is already published on isolated populations. So this really beautiful animal, this black tiger or the pseudo-melanistic tiger, is found only in one protected area in Odisha, Simlipal Tiger Reserve. This is work of Vinay, quite exceptional. We got exceptionally lucky to do this work. So sometimes you only see it here. So immediately as a population geneticist, the first question we thought about is, maybe it's because this population is isolated and by chance the mutation or the allele which causes this black phenotype has increased in frequency. So first of all, we had no clue, why does this tiger look this way? So luckily for us, because there's no way to figure that out in the field, right? I can't even sometimes see animals. Luckily for us, in the Nandankanan Zoo, there was a family which had a white mother, a white but pseudo-melanic daughter. So basically, this is not true melanism. True melanism is completely black. But here, the stripes are all messed up. Can you see? The stripes are all not regular. So this is something to do with striping. And this family allowed us to construct a pedigree, and because there was already some information from cats about which gene may be involved, we were able to do what's called association analysis in this pedigree to identify the mutation, the point mutation, which resulted in this tiger looking this way. That's crazy, right? I mean, to me that's amazing that we can actually use, we can actually figure out in a wild phenotype the causal variant. So basically this mutation is what's called a missense mutation. It converted a histidine to a tyrosine, and it was in a very conserved part of the protein. So basically we had reasonable confidence that this was the mutation which causes this phenotype. So when we went back to the field, we walked many hundreds of kilometers in Simlipal, sampled tigers, sampled fecal samples, extracted DNA, figured out which ones were from tigers, identified individual tigers, typed the mutation, right? This is a recessive trait. So only if you have 2 copies of that mutation, you look black. And basically, to cut a long story short, what we found was the frequency of this mutation. Luckily, it was the same mutation. I mean, it was unlikely to be different, but that was a relief because we've not even seen these we don't have a identified sample from a black tiger in the wild, right? We're just walking, picking up fecal samples and typing this mutation, and woof, it's there. And it's basically at a very high frequency of 60%. This is 2017, 18. So we thought, okay, maybe we missed this mutation elsewhere because you only see these tigers if they are both homozygous, right? So we looked at 400 individuals from across other parts of India. Nowhere is a mutation there. So why is this mutation at such a high frequency in Simlipal? So okay, so when we looked more at the Simlipal individuals, this is a PCA plot showing genetic clustering, and you can see that these are the other populations in central India, you know, Ratapani, Ranthambore, not Ranthambore, Satpura, Kanha, Pench, etc. Simlipal is different, okay. So it seems to have kind of different allele frequencies, and we only had 12 individuals, suggesting it's a small population. So it is small and genetically seems to be isolated. When we look at relatedness between individuals, again, the individuals in Simlipal are close relatives to each other, okay. So compared to say other populations, they are almost like their siblings or something like that. Again, a symptom of inbreeding, isolation and smallness of populations. And when we try to understand connectivity, remember we did that work where we said, okay, high traffic roads is bad and so on. When we laid those assumptions onto this landscape and tried to see what is the chance of a tiger making it to Simlipal. Well, if the tiger disperses about 200 kilometers, 0 chance it makes it there. If it disperses 500 kilometers, some small chance. And if it disperses 1,000 kilometers, it will definitely make it there. So basically, Simlipal is just small and it's isolated because it's just too far. It's really far. It's really far from any other population. So here you can see these guys, these populations are all source populations. This is our own Kanha Pench where the highway was going through. They're all close to each other with lots of individuals and lots of movement. So they stay connected to each other, but Simlipal is simply too far. And as I say that, I should just probably also say that this is actually wrong, because very recently there's a tiger which has dispersed just outside Simlipal, moving 2,000 kilometers. So these animals are super resilient. We shouldn't write them off, okay? So anyway, we just did some more work to understand how could this allele frequency be so high in just Simlipal, and these models basically show that if you went through a very strong bottleneck, very intense, very few individuals, then that's the only way you could have had such high frequencies in Simlipal. So what we're doing now is, of course, so basically the results are consistent with Simlipal being small and isolated. Human-induced fragmentation then has caused you to see a phenotype, right? It's not just that, oh, you know, fragmentation is happening. In this case, you're seeing predominance of a black phenotype, and these models projected further show that unless it is connected, it will fix. In fact, recently this was discussed in the parliament that why are there 50% black tigers in Simlipal? Nobody is talking about the genetics, but that was a question which came up in parliament. So what we're trying to do, and we did actually sample last January, we're trying to see is the allele frequency going up, and one thing which we did not test for is what's called selection. So maybe it's advantageous to be black in Simlipal, right? And that's why the frequency is high. But for doing that, we need whole genomes, we need good sources of DNA, but it's something we're working on for the future. Okay, so another very quick story of an isolated population. This is Ranthambore Tiger Reserve in Rajasthan, also isolated. We know that no individuals have left. Individuals have left, but they've just gone somewhere and come back. No new individuals have come in here for a very long time. And this is basically the team. Anubhav actually just got a job at the IASC, so it'll be exciting to see his work continue. But here again, we sequenced genomes from across India, and we found that tigers in Ranthambore are twice as inbred as those anywhere else in India. Okay, this is pretty dramatic, difference between them. And we also found that the so this is basically an index of inbreeding. The more inbred your genome is, the more number of bad mutations you have, right? Damaging mutations, potentially damaging mutations. So that means that inbreeding is likely to have some consequence in Ranthambore. What the consequence is going to be, we don't know. But what we see is what's called genetic load or high frequency of these potentially damaging mutations. So basically what we're trying now, some work which is ongoing, is to sample the past. So this is me, I look a bit crazy, climbed on this ladder. This is a trophy actually. So basically, while we blame the British for a lot of hunting, there was a lot of hunting which also happened post-independence and pre-Wildlife Protection Act 1972. And a lot of these are trophies in erstwhile Maharaja's houses. Now, all of you must have seen that 2 years ago, the Nobel Prize went to Svante Pääbo for his exceptional work on ancient DNA and archaic humans. And so the methods which we can access this type of material have really been revolutionized. So what we're trying to do is genetic time travel by sampling these individuals and ask questions like, when did the inbreeding in Ranthambore start? But this is something which is like a bit of a wild goose chase. We don't know what we'll find. The other thing which we're trying to do is, when we say inbreeding, we think about pedigrees, right? We don't think about inbreeding in terms of characterizing it from the genome. So can we actually build the wild pedigree of Ranthambore? So this is something which we've been doing for the last 8 years now. We basically follow animals. We identify mothers and cubs. We follow animals. We sample their DNA from this shed hair. We sequence genomes and we try to reconstruct paternity. And basically what we want to end up with is something like this. This is a huge pedigree of all the tigers in Ranthambore. So this is something, now we have, we've been sampling across 8 years and we have 62 individuals. So we are actually able to see, and we just have some results which suggest that all matings are either uncle, niece or aunt, nephew. So there is inbreeding happening, but sib mating we don't see. We don't see mating between sibs. Maybe that's inbreeding avoidance because tigers tend to disperse from, the males disperse from where they are born. But we are seeing a lot of mating between relatives in this population. I mean, there's no other option because they can't really go anywhere. But what we want to really understand next is what happens with this. So another thing which we're doing is we're working with zoo pedigrees where we find abnormalities because of inbreeding. So it's very curious, but in a lot of inbred cats, we find eye conditions, we find hip conditions, and we also find neurological disorders. So what we're trying to do is use what we know from humans and mice to figure out those mutations which we said they're potentially bad. Can we actually identify which ones are bad and how will inbreeding depression look phenotypically in the future? So basically we can also reconstruct population decline, and this shows us that about 60 years ago Ranthambore tigers crashed from a high number to like very low, about 10 to 13 tigers. So that's the bottleneck in Ranthambore, right? And what we want to understand by sequencing samples from say 100 years ago is the exact shape of this and what happened to those alleles. So basically what we're talking about here is evolutionary recovery. No, so if you look at the number of tigers, they're increasing. But in terms of the evolutionary variation, that is the genetic variation and the evolutionary potential for the future, it has not changed at all. It's pretty much the same. So how are we going to increase genetic variation in this isolated population? Right now we're just studying it to death, right? And sequencing genomes, spending gobs of money, just to kind of say the same thing again and again in a more sophisticated way. So the only answer is genetic rescue. That is, if by luck, individuals move into Ranthambore, or what we can do is what's called assisted migration, where we actually move individuals in here to see, to basically increase this genetic variation in this gene pool. So of course, this is just tigers, and we've just started some work on the quintessential inbred species, which are Asian lions. And I'm really excited about this. I've wanted to work on this since I was 25, I'm 50. So finally getting to work on this very charismatic, like it's a very classic example. And it's a crazy example because there were like 20 lions and now there are like potentially 800 lions. So clearly they've recovered from this impoverished genetic condition. How did they do that? So that's kind of the questions we are interested in. We've been working with Sukumar also, and we've shown some very interesting results on elephants, that basically elephants colonized from the north, and as you come south, you have less and less genetic variation, because it's what's called a dilution, right? You have a large amount of variation, some of it comes here, some of it goes down, some of it goes down, and so on. So basically, just by geography, you tend to have less variation in some populations of elephants. So yeah, so basically this is just, we sampled a bunch of lions in the zoo. It's crazy, the Sakarbaag Zoo in Junagadh has 90 lions, of which about 80 or so are wild-caught individuals. So we can actually just sample shed hair from the zoo, and hopefully we'll get to understand more about these populations. Basically, just summarize, the Earth has been severely impacted by humans, and we are witnessing species extinctions. Genetic studies can help us understand species better, and I hope they're important for conservation. But we're just scratching the surface. I wish there were many, many more people doing this kind of work. Our understanding of species still remains very, very poor. But I think there is hope. I think animals are really resilient. Like I said, there was that tiger which has moved, many thousands of kilometers and has been camera trapped outside Simlipal. But also very recently, I told you the Javan tiger was extinct, but there's a paper which has come out from a single shed hair found somewhere there, which seems to suggest that it's a Javan tiger. So what we thought was extinct, may have been hiding there, and we just didn't sample it for so many years. So that's it. This is my lab. I have the good fortune of working with a bunch of this extended lab, a lot of family, etc., with amazing set of people. And thank you so much for inviting me. I hope that was interesting. I hope I didn't overshoot my time.
Guys, anyone have quick questions?
Great talk, Oma.
Thank you.
Enjoyed it. My question is that when we talk about genetic heterogeneity,
Yeah.
are we talking about the overall heterogeneity, or can we actually think about heterogeneity in the certain genes,
Right.
which may be more relevant,
Yeah, yeah.
and that could actually make the life simpler for you to do the cloning or whatever?
Yeah. So basically that's the problem. So population genetics has always talked about distributions, right? So broad genetic variation across the genome. But what we know is that variation in specific genes is more important, and also that bad variation is also more important in specific genes. So that's what we're trying to get at, and ideally we would come up with some kind of a panel or something like that, where we could say these are red flags and these are adaptive or good variation, and you need to just monitor that, not monitor the entire thing.
So can you actually get to that by looking at the DNA from the population that has recovered,
Yeah.
and then find out where the mutation was?
Yeah. So that's what we're trying to do. So basically we've also done, and others have also done a genome assembly, which and an annotation, which allows us to not just read the DNA, but say what genes are where and so on. So that is going to happen now as we have more and more genomes.
Dr. Umar,
Yeah.
excellent talk. I'm talking about tracking of the animals.
Yeah.
And I know that tracking wild tigers is not easy. You're
Yeah.
going through their biological samples for tracking. Is
Yeah.
there a way to technologically track them? For example, see, one of your projects is Asiatic lions. You've got
Right.
90 of them in a zoo, I think is
Yeah.
what I said. Now, when they're still under capture or captivity,
Yeah.
cannot track them technologically and then let them free and then follow that? One more point is that I know people talk about microchipping.
Yeah.
And we just recently put a stop on BBMP trying to microchip the stray dogs because they're orphans.
Yeah.
They cannot be medically followed. But is there advances where you can track them without really invasive technology or some other ways so that you can track the animals?
Yeah. So basically, animal movement has become an incredibly important area, and for example, the Max Planck has a whole center which works on it, and they even have launched a satellite to monitor animal movement, through telemetry and stuff. That's not something which is moving ahead at all in India, because there's just like a real paranoia about I don't know, whatever, like the whole idea of catching animals, putting collars, understanding their movement and so on. But I think that there are some other innovative approaches. So for example, I have a colleague at Ahmedabad University who's just starting some work on lions where their roar may have an individual signature. So basically you could probably standardize that and then try to figure it out, basically do passive recorders, put up passive recorders, identify individual roars and track them that way. The other thing is that tigers are also extremely highly photographed and videographed. So if we could use some ML based approaches to look at these images and figure out which individual it is very quickly, these kinds of things, they're not automated at this point. It's all like someone looking either at pictures or doing something on a computer, but it's very old fashioned. So there might be ways to capitalize on these existing large data sets in the public domain. For example, in Instagram or Facebook also there are a lot of images, right? And we could try some of these things as an option for sure. Also, just to mention that in terms of genetic technology, there's something called eDNA, environmental DNA people are using like from water or something like that, where they can also in fact get not just what species, but also a bit of information about individuals. So this is all kind of coming up, but basically the challenge is doing anything high throughput in India. It becomes, you need a massive cluster, or you need some kind of a distributed network, and everybody needs to follow the same guidelines, and those things become challenging.
Thank you.
Yeah.
We can have one more question.
Madam, with the present technology, for
Yeah.
example, drones,
Yes.
tracking the animals, that is one way, and
Yeah.
different ways we can try it out. But one more thing is, have you seen any animal in trouble? Because trouble,
Trouble.
yeah,
In what way?
means they fell down on
Yeah. So
any.
there's a lot of active intervention on these animals. So for example, when we say we're not seeing inbreeding depression in Ranthambore, why are we not seeing it? It could also be because there's a lot of medication happening for all these animals. So with the lions, basically when we were in the zoo, I mean, probably we shouldn't broadcast this generally, but there were a lot of neurological disorders. There were individuals without a whole leg. So it's just that nobody's looking at it systematically. And I think, I don't know, maybe I'm wrong, but there are a lot of things which are cropping up, but because they are, these animals are being supplemented. There's so many cows, there's so much of food, that intense selection of natural selection, what we think about is really, they're kind of relaxed from it. There's medication, there's lots of other food, and so on. So yeah, I would say whether we see animals in trouble, definitely. Some of that might be natural. For example, when there's more individuals, there'll be density dependence and competition and so on. But there also seem to be abnormalities. And like I said, they're specific, like there are blind cubs. There's another tigress in Ranthambore with crossed eyes. So there do seem to be specific things which seem to be wrong with them.
Last question.
This is the last question.
Hi, Dr. It was
Yeah,
a nice
hi.
talk. So just one thing you mentioned to do away with the inbreeding. There is another
Yeah.
thing is assisted migration.
Yes.
So just from a animal behavior perspective, when you
Right.
do a assisted migration,
Correct.
is there is a rejection from the population?
Yeah. So that's something which I worry about a lot, because on one hand, as a geneticist, I would recommend movement, right? But then that's only one part of it. The behavioral integration of those animals is a much bigger thing. So I think that maybe, I mean, I know this may sound a little crazy, but what might be a better option is to do what's called soft release, where you basically allow animals to mate in some kind of an enclosure or something, and then facilitate natural dispersal. Because if you just drop an animal, I can guarantee you, if I drop an animal into Ranthambore today, it will get killed. Because there's a lot of territories there, a lot of territory holders, aggressive males, they'll just kill it. Right?
Because the same behavior we see in the dogs, like when
Yeah.
the dog travel to a different.
Correct. So
there.
basically, I think that maybe a soft, like a more of a soft introduction, basically supplementing the gene pool and then allowing natural dispersal might be a better option.
Thank you.
Sorry, did I go
Thank
too much
you.
over time?
I request Dr. Nagesh to convey our gratitude with a token for this wonderful
Thank you.
talk.
Thank you. Thank you. Thank you so much.
A fascinating talk. We move to our next speaker, who I no doubt is no less fascinating. We have Professor Saskya Van Nouhys. I hope I pronounced that right. Professor Saskya is a faculty member at the Center for Ecological Sciences at the Indian Institute of Science in Bangalore. She is a population and community ecologist. She is a population and community ecologist focused on understanding the roles of species interactions in the dynamics of populations. She has a special interest in parasitoid wasps and multi-trophic interactions between plants, herbivorous insects, and their parasitoids. She has worked extensively on the effects of habitat fragmentation on species interactions. Before coming to Bangalore, she worked for a couple of decades in ecology at the University of Helsinki in Finland and at Cornell University in New York. Professor Saskya will be speaking on biocontrol blueprint using ecological theory to combat insect pests. Welcome.
Okay. Let's see. You guys hear me okay? Okay. All right. So I think this talk well, it's been interesting listening to these last two talks. I only came to India about 2.5 years ago, and so I've kind of started fresh with my research. And so the things that I'm going to talk about today is I took this opportunity to talk about some new to me, well, and new otherwise, applied research that I'm doing in India. And so in the past, I've worked on long-term population dynamics and population genetics of species. And so this is kind of a different thing for me. Let's see. Oh, okay. Okay, so I'm going to talk about agriculture. And so why as an ecologist think so much about agriculture? And there's kind of two different sides, two different reasons. So one of them is that agriculture has a really broad impact on the ecology at every level on the globe. And for instance, water. So if you look at water use by humans, this beige or orange, whatever color, is water use by humans that's devoted to agriculture. So most of the water that people use, like take and use for something, is devoted to agriculture. Fertilizers. Very little of the fertilizers that we put onto crops actually get taken into the cells of the plants and then made into food that we then eat. So most of it gets washed through in the soil or gets taken up by other plants. So we're adding nitrogen, which is a basic nutrient for all of photosynthetic life. We're adding a ton of it to the environment. Greenhouse gases. It's actually, agriculture isn't the biggest player in greenhouse gases. And one of the reasons this is because agriculture involved plants. Plants are actually contributing, or actually mitigating CO2 in the environment. But nonetheless, methane production by livestock is a big contributor of greenhouse gases. Fertilizer manufacturing, application of fertilizers and pesticides, these things are contributing greenhouse gases. Agriculture has a big effect on soil quality. So loss of soil due to erosion, loss of nutrients from soils. Land use, 38% of the arable land globally, in different countries, is used for livestock. Most of that actually is used for agriculture, and quite a lot of that is livestock. So that means grazing animals. And then finally, pesticides and herbicides. So these are dangerous for the applicators, dangerous for the organisms around the places where it's applied, and dangerous in some cases for the humans that are eating the food products. Okay, so these are all kind of agricultural things. So that's one direction by which agriculture and ecology are sort of the same thing. And then the other is that agriculture functions via ecological processes. So, for example, I was teaching the ecology for master's level students, and I was thinking, oh, I could teach this whole class just using agricultural examples. So in agriculture, we're interested in climate and weather. We're interested in the biogeochemical cycles and energy flow. For example, all that nitrogen that we're adding flows from the fields out into the water and affects things. It's part of the large-scale energy cycles. Plants we're growing are primary productivity. We're interested in population growth rate. We're interested in life history and demography. So, for example, plant breeding. We're interested in making plants that grow quickly, mature at a certain time. Plants that we grow compete among each other for resources and also compete with, say, weeds for resources. And we're interested in consumer resource interactions, so pest management. And we're interested in mutualisms, such as pollination or mycorrhizal interactions between roots and plants where plants get nutrients. And so all of these things that I teach in the ecology course, I could just teach using examples from agriculture. Okay, so these two things, agriculture intertwines with what ecology is going on in the world, and then also all of agriculture is ecology. Okay, so there's lots to think about. Also, agriculture is an evolutionary process. So the plants that we eat are mostly, almost all of them are the result of plant breeding, right? We're not just eating them the way nature made them, we've bred them. So either through domestication gradually or from very active plant breeding, we've changed the mustard into a whole bunch of different vegetables. We've done breeding to make wheat, have really big grains, lots of gluten, lots of different things. So it's also an evolutionary thing. Okay, so we take advantage of agriculture being ecological, and the example that I'll be talking about is biological control. So biological control is using the natural enemies of insects or also plants, like, for example, the herbivores that eat weeds. We can use those for biological control. Using these natural enemies to control some pest. A natural enemy is something that consumes something that's a pest. And so that natural enemy could be an insect. It could also be a disease. So a biological control agent can be a disease. Okay. And so the creatures that are really the most interesting creatures around to me and have been for most of my life are parasitoid wasps. So I just want to remind you what they are. So these are wasps, Hymenoptera, like bees and like paper wasps, same group. And they fly free as adults, but they lay their eggs either in or on a host arthropod. And then they develop inside of there, and then they come out, so they develop as a parasite, and then they come out as these little wasps. Every insect species has a multitude of parasitoid wasp species associated with it. They're extremely diverse and common, but they're pretty small. We don't really notice them, but there's just a gazillion parasitoid wasps out there. There has to be a gazillion of them. Okay, so I'm going to talk about two projects. One of them is this trophic food web in managed and unmanaged systems, and then the other one is sort of developing a way to reduce pesticide use by monitoring insects using visual AI. And I just have to tell you, I haven't gotten far on these projects. Okay, so we have some expectations. These are really intuitive. so food web is, maybe I didn't say it, so a food web, in the way I'm thinking about it, is sort of the trophic interactions between species in a community. So trophic, I mean eating. Okay, so we expect these food webs in an agricultural system to be, have some characteristics. There's not very many primary part producers. That makes sense. There's the cultivated crops. There's a simple foraging environment. Most agricultural systems are kind of simple. There's not a lot of like physical complexity. There's big ephemeral resource pulses. And these are the crops as they mature because they're kind of synchronized, and also applications of fertilizers which sort of happen periodically. So there's these giant pulses of resources, much beyond anything that sort of happens in a natural system. And then in many cases there's suppressed consumer communities, so suppressed herbivores due to some kind of management, pesticides. Versus in an unmanaged setting, there's many different primary producers. There's more diverse primary producers. The environment is more complex. There are, of course, fluctuations in resources of availability, but they're not like really giant. And in general, there's no things like pesticides or equivalent. So these are different conditions. So when people are thinking about food webs, we often talk about food web complexity. And really what we mean by food web complexity is the number of different trophic levels. So who eats who, eats who, eats who, how far up do we go? And also far down. This one should have some trophic guys down there. And then also the number of arrows like going diagonally and the number of arrows between things, especially arrows like going back and forth. Like this butterfly, say, is prey to these 4 different parasitoids. So we say that a food web is complex if there's a lot of trophic levels and lots of arrows going all over the place. And we say that a trophic level is simple if there's fewer species and the arrows are going straight. So we expect that agricultural food webs are simple relative to the wild ones and that they're unstable. And the reason that we expect the instability, okay, the simple is sort of intuitive. The instability is because of that great fluctuations in resource availability. Okay. Yeah, I think this is all that I've said. And so if we expect the agricultural food web to be simple, then we also have this expectation that the natural enemies are going to be not such a big help to the farmer, meaning they're not going to control pests on plants. Okay, so we say, okay, agricultural systems are simple, but of course, just like anything else, there's a gradation of that. So some agricultural systems are not simple. So we can have a polyculture, which means sort of growing different things that mature at different times, different ways, and that makes the more complex food web. It makes it more stable. There's sort of ways to kind of engineer that, where you grow specific plants that have an effect, like the push-pull system, I don't know, in maize in Africa, which is a very engineered polyculture. And then kind of less extreme than these two is that orchards are kind of long-term. They can have pesticides and they can have sort of a simple set of crops, but the plants are there for many years, and so the resource fluctuation isn't so extreme. Okay, so I'm working on the citrus herbivore parasitoid food web. And so citrus, native to India, citrus is like oranges, limes, and also other places in Asia. So different citrus fruits have been domesticated in different places, but all sort of Asian. They're Rutaceae. There's wild relatives of citrus, and also there's sort of semi-domesticated citrus, such as curry, which is curry leaf, which is grown sort of intentionally, but also foraged in the forest. And then there's wild relatives of citrus that are really wild things, like shrub or sort of small tree understory in forests. And these are Rutaceae, relatives of citrus, and they're not domesticated at all. And so these are native. And citrus is cultivated in India, ranging from no pesticide use to very extreme pesticide use. and many of the pests of citrus globally are native to India, and few of the natural enemies are known. So one thing about the pesticide use, just that will come up later, is that lots of times people, so when you use a pesticide, you're usually supposed to have a reason to do it. Like you say, oh, there's a lot of this pest, so I'll spray. But the thing is that also farmers or land people are said, okay, you need to spray every so many months for this pest. They don't even know if that pest is there. They might not even know what that pest looks like. So a lot of pesticide use is kind of not well-founded. Okay, so the hypothesis of this study is quite simple, that there's a continuum of food web complexity from intensely managed citrus to wild Rutaceae growing in the forest. And I'll tell you the progress I've made on that so far. And then, knowledge about the potential natural enemies of these pests on citrus will provide some motivation to reduce pesticide use. If we know more about the natural enemies, then we would have some reason to maybe not try to kill them. And I'll tell you what progress I've made on that. Okay, so we're just starting. Now, we started out with the butterflies on citrus. Now, you might say, butterflies, those aren't pests. So some butterflies are pests on citrus. There are a lot of butterflies, or several butterflies, that feed on citrus. They're not the biggest pests, but they're the ones we know the most about, especially if we start looking at the natural species. So we start with butterflies, but we're actually working on all the Lepidoptera. Okay, if you just do a literature survey, for one thing, all of the butterflies get put into two species, and this is probably not true. It's probably that they didn't know which species it was, so they almost are all called Demoleus, which is the lime butterfly. And then there's a bunch of parasitoid species, and most of the data is about lime, sort of, you know, the round lime that we know. Okay, and so this is from the literature. This is like the sort of 35 kind of studies that we found that, that were showing primary data. Okay. Sorry, this is a list of parasitoid species. Much of them are unknown, and some of them are not exactly right. Okay. Then we went out onto the IISc campus, and there's a few lime trees on the IISc campus that are planted. So we're talking about 6 trees. So we spent about 9 months, we're in the process of doing that, just collecting caterpillars and eggs and pupae. And we found 4 different species of butterflies, just on these 6 limes. And then so far we found this list of parasitoids. Now, the important thing about this list of parasitoids is only a couple of them are the same ones from the literature. Like all those papers we read, the names don't match. Also, and it's what we're working on right now, except for a few of them, they're not known, we don't know what species, we can't figure out what species they are. They're probably an unknown species. So, so this list is really almost, not entirely, but all except for 2 species, different list than the list I just showed you of species. And it includes at least half probably new species. So the good thing about that is it means, oh, there's really a lot of diversity of parasitoids out there. That's cool. The bad thing about that is that it's work for us now to try to figure out what these species are and sort of make sure, see where they're known or anything, to learn about the species. Okay, so I don't think that IISc is particularly diverse. It's kind of diverse, but it's not you know, a hot spot. I think it's just that when you look closely, you find a lot more than when you're not looking so closely. Okay, and we managed to distinguish between these butterflies as caterpillars and as eggs, and then we found these, and this is not a very thorough search. Okay, so the reason, and this sort of what I just said, the reason that the literature and IISc campus don't match is because of the agricultural setting. It is more diverse in IISc. Maybe because of pesticides being used, makes there be a more simple situation. Also misidentification and just not sorting out what the whole system is. Okay. So that's the beginning of this project, which is larger, which is this sort of gradient going from a managed orchard with pesticides to organic orchard, casually cultivated lime and curry, unmanaged but unnatural rutaceae, meaning at IISc campus. Sorry, unmanaged but natural, I think I should be. And then natural forest understory rutaceae, uncultivated, just things that are naturally there, looking at the food web, the continuum, with this added kind of other place, which is the Bannergata Butterfly Park, where they're rearing the butterflies intentionally, but they have a lot of problems with the parasitoids. And so this is like the opposite. It doesn't belong here, it just isn't not on that continuum. In this case, we're finding a whole different set of parasitoids. And this is probably because they rear the butterflies that we're interested in among other butterflies, and so there's a chance for the butterflies to move to hosts that, for the parasitoids, to move to hosts that they don't normally use. So this is like a kind of a huge mix-up. Okay, so we're looking over this continuum, and we have these sort of really straightforward expectations about the differences in the food webs. And the point of that is to sort of say, this is the potential that we have for biological control if we stop spraying kind of without reason. Okay. Then, a related project. Okay, so the butterflies, not a huge pest, but representative of insects that are on citrus. The Asian citrus psyllid is a huge pest. So this is it's native, again, of India and some other places. And it's a global pest. It's not everywhere yet, but it's all over the place. And the reason it's such a problem is that it's the vector of citrus greening disease. So it itself doesn't do so much damage, but it vectors this disease among citrus plants. And that disease kills the trees. So since this disease kills the trees, there's a lot of insecticide use. And I would say that in India, and probably in most places that have this, the insecticide use is mostly due to this citrus psyllid. There's several parasitoids that are native to India, including this Tamarixia, which is significant because it's being used for biological control outside of India. So it's been imported and been used in the United States and some other places, and it works as a biological control agent. It's present in India, and we don't know anything about it. Okay, so this is the citrus psyllid. So a few years ago, with some collaborators, we worked on a really simple mathematical model. And so when people are spraying pesticides, sometimes they just do it on a calendar, and it doesn't really matter what they're seeing out there. But sometimes they monitor in their field, and they say, okay, if the insect pets gets above 3 per leaf that I've looked at, then I spray. And sometimes they're sophisticated, like they say, if I see 3 adults, then in 2 weeks I spray. Or if I see, you know, they can take into account the life history of the pest, and they have sort of a algorithm that you follow to decide when to spray. And this reduces the amount of spraying you have to do, because if you go out there and you find, oh, the outbreak isn't happening yet, so I don't have to spray, you skip a spray. And that's been true for many years, that there's been these algorithms. The weird thing is that those algorithms virtually never take into account the natural enemies that are also in the system. So if you see a lot of aphids, and you don't notice that actually 2/3 of them are parasitized, which you can actually see in aphids because they turn in sort of brown. Oh, there's a lot of aphids here, but they're 2/3 parasitized. If I just hold off and don't spray, then the parasitoids will take care of it. So in many cases, if you monitor the parasitoids as well as the pest, then you could reduce the amount of spray. So we wrote this model where we just looked at the effectiveness of the pesticide, the cost of the pesticide, the yield of the crop, the mobility of the pest, and the mobility of the natural enemy, and how hungry the natural enemy is, all these different factors, and kind of made a bunch of plots just showing how you can change the number of sprays that you need depending on the natural enemy and the pest. And so all we did was sort of take models that exist for the pest and add the natural enemy, which seems like a weird thing that hasn't been done, but it hasn't really been done. Okay, so we wanted to do that for the citrus for the Asian citrus psyllid. The problem is that it depends on being able to monitor both the pests and the natural enemies. So the modeling is easy, but to actually figure out the natural enemies, it's very hard. So I'm like a parasitoid biologist, and I look at these yellow sticky traps. This is a yellow sticky, so I look at these yellow sticky traps, and I can find the Asian citrus psyllid, but I don't know what any of these other things are. Some of them are parasitoids. I've probably even worked with them. I can't tell. They're like, so they're in perfectly good shape. Like, their bodies are all there, but they're all contorted, and who knows what they are. So you can't expect a farmer to know all of these things. Like, parasitoids all basically look the same. Okay, so the plan is to train AI to recognize the particular species on these sticky cards. So you put these sticky cards in the field. The insects are attracted to yellow. Most insects are attracted to yellow. They fly there, they land on there, and then you scan this card, send it to the AI, and they go through this algorithm, and then there's a part in there, which is what we're working on right now, where there's humans who are training the AI. So we're going and saying, that's a Asian citrus psyllid, that's a citrus, blah, blah, blah. We train it, it tries, it gets a lot of them wrong, it misses a lot of them, go back and train it, and we're doing that with the citrus psyllid and then 2 of the parasitoids. And so at the moment, we're actually doing really bad. We're doing okay with the citrus psyllid. We've almost got it to learn the citrus psyllid, and we're doing awful with the parasitoids. I have never even seen one that I'm sure is the parasitoid species. So working on that. So the point of this AI thing is, in one sense, it's to learn the abundance of the parasitoids in Indian citrus. Like, we know this useful parasitoid exists and it came from here, but we don't really know whether it's in the relevant places or not. And then the bigger picture is to use it with that model that we've made and try to reduce spraying. We're also working on it to, if we can put these cards at like ports of entry into places where the citrus psyllid hasn't yet come, like Israel, then we can use AI to scan these cards, and then you can see whether it's coming in at ports of entry, and it's just sort of a cheaper way of trying to catch an invading pest. So we're also developing it for that. Okay, I think that's it. Okay, so that's it. So it was these sort of 2 illustrations of work in citrus that's sort of merging ecology with kind of agricultural questions focused on biological control. And so for this project, there are lab members that are involved, collaborators here in India, and then collaborators in Israel for the AI sticky trap thing. That's it.
Do we have any questions?
Sorry, just a quick question. Why is the AI worse with the parasitoids? Is anything to do with the fact that they're smaller, or?
I think there's 2 reasons. One of them, they are a little smaller, but they're not that small. And it's not because they're smaller. It's because there's a lot of things that look like parasitoid. Well, there are some other parasitoid species, and then even the flies, like flies that have kind of long legs, they end up looking a lot like parasitoids. That's why. It's basically they have long legs like flies.
So, thank you, Professor. Very interesting talk. You talked about 2 things. One was your objective, which was biological control of pests.
Yes.
And then you talked about the complexity of food webs.
Yes.
As it gets more complex.
Yes.
When you leave them in nature.
Yes.
My question is, I'm trying to understand the direction of causality. Are you saying that the objective is to introduce species which take care of pests, or is the objective to increase the complexity of the food web which will lead to pests being taken care of?
Okay, yeah, that's good. Both. So like biological control often gets divided up by those two ways. So one of them is like where you bring the missing species. For example, the parasitoid of the citrus psyllid has been brought to Florida. That's one kind of biological control. But then the other kind, conservation biological control, is increasing the habitat complexity in order to build up the existing natural enemies. So they're both called biological control, but they're really different. Yeah.
Any more questions? We can have a few more questions. Anyone has any questions?
So I will go one more on the AI part that Dr. Uma asked. I think one of the biggest challenges would be that we wouldn't have enough training data of previous records of the insects being labeled, and when the humans are doing the labeling part. And
Right.
Dr. Uma talked about using eDNA in her work.
Right.
Is there an approach to use eDNA to create the labels which can then be used for training the visual system, which is much more applicable and scalable?
Right. It could be. I mean, when we started doing this, we thought we would be able to see the parasitoids well enough that it would be more efficient to do that by eye. Because if you do the eDNA, I mean, it would be actually taking that card and kind of dipping it in, I don't know, or scraping it or something, and there would be so much other stuff. I mean, the cards have hundreds, if not more than 1,000 creatures on it. We thought it was kind of overwhelming. But now we actually have moved to thinking about eDNA because we're having trouble with by eye identifying them, and so that's taking a huge amount of time. So the by eye one, it just takes that's the link that takes humans. Like so far, we need humans to do it, and so it could, yeah.
Yeah, I meant only for the training of the AI
Yeah, right.
algorithm in the lab.
Right.
Once it goes in the field, then it's all visual.
Yeah.
But for the training of the...
We thought about it. It might make it much faster.
One question here.
Yeah.
Yeah, the overall objective is agriculture and increasing crop productivity with less problems, right?
Yeah, or at least,
So it's a very nice talk. I
Yeah.
also enjoyed listening about parasitoids, natural controls of our enemies.
Yeah.
My enemy's enemy is your friend.
Yeah.
Right, so like that.
Yeah.
And but I think there's the other one which I'm sure you're aware of. I'm just wondering if you're also looking at that in terms of pheromone control of the pests in themselves.
Yeah. Yeah, you mean another, yeah. I have actually done some work with pheromone control, but not this, I don't happen to be doing it right
Not
now.
yet, okay.
But yeah, I mean, that's another way. Like, for example, in the push-pull system, it's using, it's using the odors of unattractive plants to mask the odors of attractive plants. And so that's a whole different, that's a whole different sort of modality of using ecology for control. But yeah.
Thank you.
Yeah.
I request Dr. Praful to come and present a token of our gratitude to Professor Saskya. We will now break for lunch, and we will resume at 1:40.